Alright, welcome back. We are talking about DNA in our genes and our essence, the essence of life. Okay, we have Prof. Dr. Zafrina Zainuddin. Prof, we have been having a very interesting conversation. We have to go into a very controversial topic in that sense, which is gene editing.
Alright.
So, gene editing, it's been talked about a lot. There's been a lot of conversations. Everyone is talking about it. What does it actually consist of?
Okay, basically it's just a very simple method on to change the sequence of a gene,
Okay.
to change or either to change a single mutation to the one to a better one, or to change the mutation that causes a disease. So for you to obtain the preferred phenotype.
Oh, okay.
So basically there are a lot of methods, but currently CRISPR is one of the in things.
It's the in thing
the
right
in
now.
thing,
Okay.
yeah.
We have to talk about the conversations. There's been a lot of talk about the gene editing thing because there's a lot of fear.
Exactly.
The fear of the unknown. What are we actually capable of? We now see in movies, you know, people are talking about gene editing. A monkey is as big as a building and things like that. So people don't really understand where we are exactly in that sense. They say we are actually playing God. So what is Prof being in that, doing that gene editing? What do you say about that?
Okay, theoretically, if we have a method, the method has been proven to actually, and the method is capable of changing the DNA sequence. Theoretically, it can happen, but practically, I'm not sure whether how successful it could be. So like the Chinese scientists has actually claimed that they have a twin edited gene babies have been born. So is it true? So
it's a claim.
It's a claim. So because he has actually violated all the procedures in the lab and everything, the ethical clearance and everything. So we can actually how to validate that, how to make sure that it's true. But theoretically, it is something very scary. If people can actually change a human, a natural, what God has created, do you like to see 10 people having something exactly what you are currently having? So do you want to see another Usain Bolt, okay, winning an Olympic medal, but knowing him is as a genetically modified organism? Is it fair, socially fair? So I think there are so many things to actually talk about when we talk about gene editing. But one thing for sure, if we can actually edit the gene that causes the disease, we can actually, this genetic engineering is something that is huge. But if we can actually focus into to cure a disease that actually caused by a single gene mutation, that would be great.
Yeah, there has been a lot of talk about doing that. One of the reasons gene testing, or actually gene editing,
that was the main.
That was one of the main reasons. Yeah. Like talking about the playing God and things like that. Do you think are we very far from actually creating a human being with genetically modified human being? Are we very far from that? Are we like 50 years behind that, or is it not as far as we think?
Theoretically, no.
Okay.
So because the method is already there, is this how whether it is going to be a positive, whether it's going to give a positive impact or a negative impact. But theoretically, lab-wise, method-wise, the method is already there. The method has been established. So how are we going to use it? This again is the question, because like what we mentioned just now, CRISPR is actually is a DNA strand. It's a strand that is actually being created by the bacteria to protect itself against the invaded virus. See, so it's for its own protection. But as a human being, we are so creative. So now we are using it to actually change, to make our strawberry more sweeter, and then we are changing our chicken so that we have a bigger chicken, or a chicken that are actually less prone to diseases and everything. So how we use the technology actually.
Okay. And CRISPR is actually quite interesting because when you read about CRISPR, a lot of people talking about it, it's actually, there is a memory in that sense. You actually know what viruses actually have
yeah, based on that
attacked.
short sequences that has been actually kept by the bacteria, so we can actually know what are the virus that has actually attacked that particular cell. That is actually, it's a mechanism of protection.
Okay,
Yeah.
all right. And moving on, other uses of CRISPR, we can talk about disease. How does that can be used, like fighting off diseases, or maybe can CRISPR be used or gene editing be used to find a cure for like rare diseases and things like that?
Yeah, I think there's one clinical study. They will actually they have injected these cells that has been modified or engineered using this CRISPR method into human. I think that has been done some time back in 2017, but I'm not sure whether it was successful or not. But that particular study was actually to cure a disease. So there's also one study, I went to one of the conference, that particular presenter, he said he has actually successfully cure a boy, a 4-year-old boy, having this muscular disease where the boy actually, when he gets older, he will actually, all the muscle will get weaker.
I see.
If I'm not mistaken, it was Duchenne muscular dystrophy. So he did this some genetic engineering, and he put it back, the cells, into the boy. And after few months or maybe 1 year, the boy starts walking. So the photo that he showed to us was the boy was actually climbing the stairs from not being able to do anything. So there are the good things from this genetic engineering, but again, how you want to use it.
Okay.
Yeah.
All right. Maybe the final question: what can we actually expect for the future? What actually do you think, maybe in 10 years, 20 years time, that this genetic engineering will actually help?
My fear is actually to those who created this all biological weapon. Yeah, that is my fear, where people abuse the knowledge, the method and everything to create something that is can cause disaster. But for curing disease, I can see that there's a lot of potential that we can cure this beta thalassemia that is actually caused by a single mutation in the gene. Because we have a lot of cases of thalassemia in Malaysia. I have 2 cousins, they die at a very early age because of thalassemia as well. So precaution is good. We have all this screening, all this gene screening, right, for thalassemia. But in future, like in 20 or 30 years, if you can go to a clinic and give your blood or your cells for a genetic engineering, and after that, after a year, you are cured, that is a miracle. That is what we are actually looking forward to, rather than, but my fear is like there are more people actually abusing the technology rather than actually doing it for good.
But all we can do is just hope for the best.
Yeah, hope for the best.
All right. Thank you, Prof, for joining me today, and I appreciate everything that you've said. If you want to watch this back, please follow us on all our social media platforms and also the Ashrawani app. My name is Dishan Kumar. Thank you for watching. Goodbye.